my pleasure to invite to you this to deliver the talk on synthetic biology revolution, past, present, and future. Now, the mic is over to Dr. Sanjay Ghosh. Welcome you, sir.
Thank you very much, Professor Balakrishnan. So I must thank you first for a very kind introduction, very generous introduction, and also giving me the opportunity to come and give a presentation on the subject of synthetic biology. I do think that synthetic biology is a fast evolving and an upcoming area of biotechnology. So I really congratulate you and your team to organize such a event where to mostly excite the people in India, especially, to get involved in synthetic biology approaches and to get involved into practicing synthetic biology, which we all can do in our own capacities. So I really thank Professor Balakrishnan and the university for the invitation and the opportunity. So let me first try to share my slides and then we can get started probably. Okay, so I'll just put a pointer somewhere. Okay, perfect. So yes, so as you all know that synthetic biology is a very vast field. So today what I'm going to do is to touch upon how it all started, what are the basic principles for synthetic biology approaches, and what is the potential of synthetic biology in the future. And I must say that more people get involved, the better this field will be, because it's quite in a nascent stage. So I hope at the end of the talk and throughout this event for the last 3 days, ending tomorrow, you all will be a bit more excited and will think about using synthetic biology projects in your own places. So just to get started, first let's see what synthetic biology means. So there is not a single definition available for synthetic biology at the moment, but what broadly it includes two different criteria. Number one is that either we can make completely new biological parts which doesn't exist in nature, and for that we need to engineer DNA elements, genetic elements, etc. Or we can redesign. So there is a natural biological system, let's say a cell. We don't have to create a cell, but we just engineer that cell to suit the purpose. And mostly synthetic biology is done for useful purposes for the humans, right? So it is no fun engineering a cell if it is not going to give us any benefit. So at the end of the day, synthetic biology is a combination of biology and engineering principles. So of course you don't have to have an engineering background. I don't have, but at the end of the day, we can take the basic principles of engineering discipline and apply in the biology. Now, as you know that, for doing that, we need to understand the biological system well, and we still have a lot of work to do in terms of understanding even how a cell functions. And without knowing that, we cannot engineer, right? So that is the main challenge currently the synthetic biology has. The take-home message is that synthetic biology is the engineering of biology to generate functions that do not exist in nature. So normally in the natural conditions, in the natural population, naturally in any cell type, those systems don't exist. And anything that we engineer into it for useful purpose is included within the area of synthetic biology. So you can imagine it involves a lot of things: genetic engineering, metabolic engineering, etc., etc. And it is also a very multidisciplinary field. So it involves people working in computer science, biology, engineers, clinicians, etc. So it is a kind of a melting pot for different branches of science to come together to be able to engineer a biological system. Okay. Yeah. Okay. Sorry. So, what are the basic principles of synthetic biology? So, just like we make an automobile So these are engineering principles, right? So we have parts, nuts and bolts, and then these nuts and bolts and different engineering parts are assembled into a device, let's say the engine and all other components, and this entire device is then put into a chassis, right? We put an aluminium cover on it and it becomes a automobile, in this case a car. So in the similar way, the synthetic biology also uses these similar principles. So we have parts, that means we have the genes which will translate into proteins, and those genes and proteins are the parts. Then these genes can be assembled into a given pathway. That means one gene can switch on or regulate other genes and thus create a device which will give an output. That means if we give a input at one end, we should get an output at the other end, which is easily controllable. And such devices are then transplanted into a host cell. In this case, I mean, the widely used host cells in synthetic biology are bacteria and yeast, because those are simpler cells. So they are kind of easy to engineer, and a lot of biology is known about them. So at the end of the day, we need to develop a system where we can give an inputs, in this case, let's say carbon source and any kind of environmental stimuli, and we get an output in terms of either a medicinal purpose, let's say metabolites, which are useful for humans, or even use this engineered cell for sensing any kind of ligand, and it can be used as a biosensor as well. So it has quite similarity, as you can see, between the engineers, what they do to, let's say, manufacture an automobile, and what biologists do in the labs to manufacture a cell or create a cell which will give some applications for the human use. So now, what is the workflow? How is this done? So there are several steps. The first thing what one has to do is to find a target. That means, what do we want to engineer? Which protein do we want to express? What kind of metabolite we want to synthesize? So that is the first target we have to identify. This is usually done by bioinformatics based approaches, and it is heavily dependent on the knowledge that is already generated till now. So this is not a basic research, but we are using the knowledge generated by the basic research to design or to identify a target. Once the target is identified, and then we come to the parts. That means we have to design the genes, whether these genes need to be codon optimized depending on which host we are going to use, etc., etc. And then all this restriction site mapping, nowadays there are softwares which easily do these things. After the gene is designed, then which vector to use, because the vector has to be host specific. So there are prokaryotic vectors, there are eukaryotic vectors, which will allow expression of genes and regulation of genes in a given host. And in this case, when we are expressing something, then we need these features. We need a promoter, we need a ribosome binding site, a transcription terminator, a selectable marker to select the engineered cells, and also this plasmid should be able to replicate within the host, so we need a replication origin. So these are the features that we need to integrate within the vector in addition to the gene, or for this gene to be expressed in a heterologous organism. Once we have done all of that, then the question is, how do we integrate into a circuit or a pathway, right? So in that case, either we can use a natural pathway or an engineered pathway. What does natural pathway mean? That means we can express a series of enzymes or a pathway which already exists in plants, but let's say I want to put it in E. coli. So that's a natural pathway. I'm not changing the genes at all. I'm just optimizing it for expression in E. coli, but that pathway exists in nature in another host, and I'm just putting into a different host. And in engineered pathway, we can make de novo, like in silico genes and their functions based on what knowledge we have, and it is completely synthetic. That means that gene or that function doesn't exist in nature before that. So this pathway assembly is crucial before we put it into the heterologous host. And this host are, as I said, are mostly bacteria, yeast. There are good examples of algae and other fungi being used. So once you make a recombinant organism harboring these pathways, which carries those genes and the vectors, then they are grown into fermenters, and that desired molecule or the protein is made in large quantities. So that is what biotechnology is all about, right? And then these are, of course, have to have some uses. And those uses can be that these engineered organisms make drugs, metabolites, fuels. Like it's a very upcoming field is that we want to cut down carbon emissions, so can we make biofuels? And then also therapeutic proteins. There are a lot of drugs, protein or biomolecules we use every day, such as insulin, which is made by yeast. It is not derived from the animal sources at all. So these kind of applications are possible and are now quite widespread using the tools and the workflow of synthetic biology. Now, what the workflow, like how this is achieved. So, as I said, what we need is to engineer a cell where we put an input and we get a desired output. And then we decide on the genes, the promoters, the host organism, and all those kind of different things. So all of this, we have to first step is a design. So in the lab, we first have to kind of go to the drawing board and think what we want as an output and what is an input and what kind of genes will take that input and make it as an output. After design, then we have to build cloning, DNA, cutting, pasting, making recombinant organisms. So this is all we are building the organism and genetically engineering after design. So this is on the work bench, like in the lab bench, so-called wet lab work. Then we have to test whether we add an input and we get the output or not, because this is heterologous. This we don't know whether it will work all the time, although we have to design carefully so that the chances are high that it will work, but at the end of the day, we have to test. And if it tests and it may fail or it may work, from that we learn. That means maybe the output is not as robust as we would have wanted. So we have to then change something into the system and go back to the design again and again tweak that pathway and test again. So this is a kind of a circle of steps that are required for the synthetic biology output. Since we are doing it mostly for the first time, or maybe we are optimizing something, so this cycle is kind of iterative. It will go on one after another unless we get the desired output from the input that we intend to in the first place. Now, what are the different genes? So these are the main components. So as like nuts and bolts in a car, we also need standardized parts. Now, for standardized parts in a synthetic biology project or in a system, we need these different genetic elements that you all are aware of. So we need promoters. That means these are the sequences of DNA which control the gene expression, transcription. And then there are protein coding sequences. There has to be terminators, etc. So in the prokaryotes, the architecture of a gene is relatively simple. However, in eukaryotes, there are different insulators, enhancers, etc., which control the gene expression. And this is very important in synthetic biology, is to understand what are the controls of gene expression in a eukaryote versus prokaryote, because that will decide what kind of promoters, what kind of genetic elements you need to engineer in your vector so that you can control the biological system in a very robust manner. So these are the different components, the pieces and parts that is common to all synthetic biology workflow. Now, this is a bit loud slide, a bit busy, but here just the take-home message is that in eukaryotes, the promoter itself is kind of quite complicated and we don't understand fully, and there is no a kind of a consensus sequence for that. So there is definitely a core promoter that we all are aware of, which is just a Tata box and some -35 sequences, etc. But this promoter is regulated by a lot of sequences upstream of it, which can be either close or very far. If it is close, then it is a proximal promoter sequences and factors that bind to it, or it can be very far away in the chromosome or in the DNA. So these are distal promoters, and they are not always mapped. And they can have either a positive influence or a negative influence on the promoter. So when again we are engineering, we have to choose the promoter in such a way that in principle it should have less of an interference from other parts of the genome, because we don't want any kind of feedback more than what we have designed for, right? And these are the different cis elements that are there, and these cis elements must have been characterized, that which are the transcription factors which bind, what are the repressors and activators that can bind to such sequences. So these kind of cartoon shows that a promoter is not so easy to design, and there are variety of regulatory elements that we have to keep in mind, especially in terms of when we are expressing something in eukaryotes. Prokaryotes is a bit simpler. Now, what are the different types of promoters? So, why it is important to know is because the heart of most gene expression systems in synthetic biology or otherwise depends on promoters. Whether we want to overexpress a protein, whether we want to control the expression of a gene, they are determined by promoters only. So these promoters are of different types. As you might know, I'll just quickly go through it. So there are constitutive promoters where these promoters are always firing in all cell types at all the times. So they are not kind of not very tightly regulated, and they code for housekeeping genes. There are inducible promoters. That means these promoters are off in a given situation, but they can be switched on when they receive some environmental cues. They can be chemically or physically regulated. Then there are some tissue-specific promoters. That means in multicellular organisms, in eukaryotes, we have the promoters which will fire only in a given cell. So let's say only neurons, nowhere else in the body. So those are tissue-specific. And then there are completely synthetic promoters. That means we can take pieces from these different types and we can make our own promoter which we want to regulate the expression of a gene. And this is what is kind of an it's an upcoming field where we want to work on to find synthetic promoters. Now, why synthetic promoters are important? Because we want to control the biological system in a very defined manner. If we have these kind of promoters, then we don't know these upstream sequences may regulate them when we don't want them to. So there is a huge amount of research going on, and even in my lab where we are trying to make synthetic promoters which are sequence and context independent. In principle, if we design a synthetic promoter, it should be used not only in yeast, but also in mammalian cells probably. So that's the race currently, is to do standardized parts in terms of promoter activity. Now, these are the kind of cartoons which basically show that how to organize a promoter. So we always used to, if we're making a synthetic promoter, we need to keep the starter box and a -35, which we cannot change. And that is a minimum core promoter. And then these are the cis-acting elements depending how we want to regulate the promoter. And of course, to measure the strength of the promoter activity, we always put a reporter gene and a pollination signal and other genetic elements. So this is typical in mammalian synthetic promoter, how it is done. In terms of prokaryotic synthetic promoter, it is quite easy. It's easier. We don't need all of that. We just need to know that we have a data box, we have a -35 element, and these lengths, these distances are important, but the sequence may not be, and that can be changed and made completely synthetic. Now, how to do that? So one of the ways, these are some of the approaches that people have done. Let's say this is a CMV promoter from the mammalian cells. We can take the data box of that. So that is the core promoter. And then we know from the literature that there are certain transcription factors which bind to certain sequences. So now we take those sequences, fuse it to this minimal promoter. So this promoter sequence now doesn't exist in nature, so it is synthetic. And we have designed in such a way that these sequences will bind to a given factor, a transcription factor. So this transcription factor and this sequence interaction must be known beforehand. And that's why synthetic biology is mostly knowledge-based. So what information is out there that we can use it for translation purposes. And if this information is not there, then probably we cannot make this kind of promoter. But since there are several reports of gene control, we can make either unidirectional or even we can make bidirectional. That means we can regulate two different genes in two different directions. So the opportunities are endless, provided what kind of well-defined characterized parts are available. So this is another example where we can control the expression of a gene just by putting two different sequences of promoters which bind to two different transcription factor complexes. So it can be triggered on, let's say, by two different signaling networks, because the signaling networks always lead to phosphorylation of a protein, and that protein goes to the nucleus, binds, etc. So we can engineer those binding sites upstream of a core promoter, a minimal promoter, and then regulate this transgene expression based on a signaling. So if we add glucose, that let's say, then because of this MAP kinase signaling, it will go and trigger this, no other time. Right? So these kind of small circuits or on-off switches can be made, provided what information do we have in terms of DNA sequences which bind to a transcription factor, and which transcription factor is regulated by what signaling. So you can imagine we can have a cell where we can adding some environmental or external cues, chemical or physical, we can control the cell behavior because we are expressing certain genes in the cell ectopically. So how they are discovered? So there are a couple of papers out there that released. These are the minimal core elements, and these distances are known. So people have randomized this, and then they have done a screening. I'm not going into the detail of that, but just to say that a lot of DNA oligos are made, and then they are screened for promoter activity, and they found that typical promoter in yeast is around 500 or 600 nucleotides or base pairs. But in this case, they finally ended up in a promoter which is a core promoter and as strong as the strongest promoter in yeast, and the length is only 150 base pairs. So you can see that you can reduce the size of the promoters, and it is synthetic. So, and it is as strong as a native promoter. So these are kind of advances that are needed to make these standardized parts. Now, what are the advantages? Of course, they are programmable because you can take these elements and put it somewhere else. They are predictable because they are not to be regulated by any host factors. So that will mean that whatever gene you clone downstream of it will be reproducibly expressed. So every time you use this promoter, you will get only that much expression. And of course, as I emphasize, that these promoters can be very small, and this allows to make multi-gene pathways. Because if the cloning becomes too cumbersome, because the size of the DNA is too big, we cannot clone easily. So the smaller the size of these elements, more genes you can clone in a vector. So that gives an advantage of making synthetic systems. Now, let's come to the part where how all of this started and what are the technological advances which made all of this possible that we are talking today. So, if you look at the timeline in 1960s, Jacob Monod, we all are aware of this lac operon, right? So he found that in principle, in absence of lactose, this lac operon is absent because there is a repressor which goes and binds and prevents the RNA polymerase from transcribing. However, as soon as you add lactose to the media, then this repressor is inactive and these enzymes are made. So this is a classical example which got people thinking. That means we can make a regulatory circuit in a cell which can be controlled just by adding a chemical outside. And that was the kind of a birth of an idea that we can regulate cells if we use these sequences and repressors to control the gene expression. And here, this example being how a cell responds to the environment. In the environment, if it doesn't find lactose, it is off. If it finds lactose, then it is on. So there is a on-off mechanism. And this was done in 1960s by Jacob Monod. So similar to that kind of system, which is also used in synthetic biology a lot, is UAS Galfor system. So this is a yeast-based system. So in yeast, just like in lac operon, where they use lactose, yeast grows on glucose, but you can also, when you add galactose, then a different set of genes are switched on. So when there is glucose, those genes are off. When there is galactose, those genes are on. How does it work? There is a transcription activator called GAL4, and this GAL4, when there is glucose and no galactose, is sequestered by this protein GAL80. And because of this, it cannot bind to the DNA and no transcription of these GAL genes. The moment we add galactose and not glucose, this will lead to transcription and translation of a protein called GAL3. GAL3 will go and bind to GAL8 and will release the GAL4. Now GAL4 is a transcription activator. It is free to bind to UAS sequences and switch on transcription. Now, how to use this for synthetic biology purposes? That means we can spatially and temporally control gene expression based on this two-component system. And one of the examples is that we can take this GAL4 protein gene, we can clone it under a promoter, which will be triggered only when we give heat shock. So we make a cell which carries this transgene where the promoter which drives GAL4 is inducible by heat shock, a physical stimuli. When you give a heat shock, then the promoter fires, GAL4 is made. Now this GAL4 can go and bind to this UAS sequence, which is engineered upstream of a promoter, and your favorite genes get active. That means in absence of heat shock, your protein, your favorite protein is not expressed. When you give a heat shock, it gets expressed because of this two-component system. So that's how that gives rise to the idea, and it is kind of based on the Lac-Operon system, that you add an inducer or you don't add an inducer. A gene is off and a gene is on. So that helps in making the synthetic circuits that we'll come to. The other thing that was a technological advancement in 1970s and 80s, we know that in 1970s the recombinant DNA technology started picking up pace. So people could take foreign DNA, clone it by restriction enzymes into vectors, and then they can make the recombinant bacteria, and which can be selected quite easily. So that allowed foreign DNA to be cloned, right? So this is what recombinant DNA technology was picking up in late 70s, and in 80s, Carey Mullis devised the PCR. So now, not only you can just take a foreign DNA, you can make millions of copies of that, and then also clone into the vector. So this was a huge technological leap that meant that we can clone, we can amplify and clone any piece of DNA into any foreign vector. So this was around a decade where tools, technologies, and methods for genetic engineering were kind of invented, and people were cloning, making recombinant cells quite easily. So this allowed, hereafter what happened in 1990s was the advent of the sequencing technology. So earlier it was the Sanger sequencing, which was quite cumbersome, but then came the automated DNA sequencers. And these sequencers could sequence huge amount, like a very high throughput samples. And these, what we are now very commonplace, we always do this Sanger sequencing to these companies who use these fluorescent dyes, and they can sequencing millions of bases in a given day. So that also helped, this technology actually helped and enabled to sequence genomes of organisms. So if we look at the sequencing technology that was developing during the 1990s or even around earlier 2000s, we had the first generation DNA sequencing, which was Sanger method, but modified for fluorescent dyes. Then came capillary electrophoresis around 1990s. Then came this so-called second generation massive parallel sequencing, which is the next gen sequencing. And it started with the Solexa 454 in the mid 2000s. And towards the end of the decade, we had very high throughput third generation single molecule sequencer available, which is currently in use for sequencing the entire genomes. So that means the technology was available by 2010, where we can take any cell literally and we can get the genome sequence within a week or so. And this was possible because of this evolution of the sequencing technologies. So if we look at this timeline around that time, the first genome to be sequenced was for Mycoplasma in 1995, but then there were a variety of genomes being sequenced and the technology was being developed, so that now at the end of like around 2012, we have around 1.5 million organisms or genomes available in a database. What has happened because of this sequencing technology is the cost of the genome sequencing has gone down dramatically. As you can see, it was a huge amount to start with per genome. Now it is less than $1,000. So anyone can sequence any genome, literally. That also led to availability of complete genomes quite a lot. So you can see the graph, it picks up around 2002-3 when this automated DNA sequencers came, and it has exponentially increased over time. So that led to availability of huge amount of genome data in the hands of researchers. At the same time, when this genome technology was being developed, there were high throughput technologies were also there to measure proteins, transcriptomics. So the same sequencers were used to measure the transcriptome of an organism or a cell. Proteins, mass spec, and high throughput methods to quantitate lipids and metabolites. So that means now we are trying to understand, we had data to try to see in a cell what are the genes that this cell has or this organism has, how at a given time, how much of it is transcribed, and this transcription leads to what kind of protein, lipid, and metabolic profile. So there was a huge leap in the 2000s to get the kind of a holistic view of a cell. How does it work? And we need to understand the biological system before we start manipulating, right? We need to understand how an engine works before we change it to make a better fuel efficient or hybrid engine. So this was a turning point, in my view, in the 2000s that these technologies came, which enabled synthetic biology to start and pick up pace very rapidly. So now, since there are so many omics data, metabolomics, transcriptomics, genomics, proteomics, so there was a huge amount of data, and that started the bioinformatic revolution, right? So all this data has to be made sense of. So that will attract a lot of computational biologists. I mean, that's the computational people who came to biology, and they were basically analyzing all this data to form new hypotheses. The biologists were, testing them in the lab, and then getting new insights: how a cell works, how a gene is regulated, and if you knock out a gene, then what kind of global changes happen within a cell. And that was very important because then you're understanding how the system is perturbed if you just knock out a gene, right? And these raise new questions, and these new questions means that we need to analyze even better. And so new technologies were developed, and this was a cycle that went on from the 2000s, where different people with different expertises, computational biologists, and even technologies, they came together to work in the area of synthetic biology. And this was the start of synthetic biology. So that gave rise to the first kind of a broad area, which is called systems biology. It's still quite popular, and it is still very important to be able to understand the biological system. Because what systems biology does is takes this high throughput data, all omics data, and then try to analyze and make a model, and different gene networks, how are the protein expressions are related, which protein regulates how many other genes, etc., etc., and then feeds it back so that the experimentalists can check it and can verify those models, and then again build. So this is again a kind of a build, test, learn model it is going on. So systems biology is mostly an in silico work done by the computational biologists, but this is very important in terms of understanding the biological system, which is central to manipulating a cell or a system for synthetic biology purposes. And this all happened in the decade of 2000. Now, the systems biology, one small example, we have all this multi-omics data, proteomics, transcriptomics, etc. And then these systems biologists could actually identify some markers. They could take a disease cell and a normal cell and try to see what changes happen in a disease cell. And thus one can identify markers. That marker can actually lead to personalized medicine. That led to the realization in 2000 that the same drug may not work for all humans because we are slightly different. We have SNPs, etc., and different people react to drugs differently. So that kind of gave birth to the idea of so-called personalized medicine. And then once we know that we need personalized medicine, we need to have this drug which will be targeting specific genes or genotypes. So genotype phenotype thing was completely changed because of this particular area coming. And this led to also an important thing: how can we manipulate biological systems in a rational manner? That means not just doing a screen, but we know which gene may affect or control which other genes. So can we make a drug which will knock out that central factor and thus not lead to a given disease? So this was very important because synthetic biology at the end of the day, even now, is rational manipulation. It's not just hit and trial all the time, but where we know what we want and then we rationally design all the vectors and gene systems. And for that, as I said, it is knowledge based, and the knowledge comes also from systems biology approaches. So this all lead to the whole idea that a cell can be seen as a electrical device, let's say, right? So that means we can give an input, there can be sensors, and these sensors by this protein phosphorylation and other gene networks, which we understood by the time in a global manner, can be integrated into these so-called logic gates. And these logic gates can give an output based on which there can be transcriptional networks, and the transcriptional networks are heavily dependent on the type of promoters, right? So what kind of promoters will give what kind of output? So these were then integrated together so that the output is what we want. And in principle, we can have a lot of different outputs. So this was the start of synthetic biology, which picked up pace in the year 2000s. Now, the first thing that came completely synthetic and kind of going towards this automobile manufacturing is to make switches. So we make an on-off switch or a toggle switch in electrical devices. Can we make it using genes? And the answer was yes. So in 2003, the first report came where they designed and validated synthetic gene circuits. And this was based on inducible promoters. So let's say there is a lac repressor and there is another repressor called CI. So CI is going to block lac repressor expression. Lac repressor is going to block CI expression, right? Now, if that is the case, then the GFP, which is now here, is flat. I mean, it is low amount of GFP. The moment we give IPTG, IPTG will inactivate lac repressor that we know from the lac-operon. If it inactivates the lac repressor, then CI will be activated. So then that will lead to more of GFP. Now the system is going into a stable of GFP. Now at the moment we add heat, heat will activate this repressor. If this repressor is off, then lac repressor will actually switch on and it will inactivate GFP. So you can immediately see that the GFP expression falls back to the basal level. So that means we can make, these are called toggle switches. That means there are two conditions where you can toggle between and the system remains on the same plane as long as the second stimulus is not given. So this was the first design to show that biological systems can be manipulated using synthetic elements and they can be maintained by just adding externally some cues such as IPTG or heat. There were also autoregulatory circuits designed. I'm not going to detail, but it just shows that one can use a repressor to repress its own expression and thus have a very sharp peak of gene control. So instead of a very broad leaky expression, one can make it sharp just by expressing a repressor which represses its own expression. And this is a biological negative autoregulatory circuit, right? It negatively regulates itself. So these were all published in around 2003 and 4, which around the time when this DNA sequencing was being done and genome was being read. So that was the era of genome reading. So around the same time in US mostly, they started the first synthetic biology conference and the center was established in US at MIT. They were the pioneers in that. They brought engineers, computational biologists, etc. under one umbrella and they started this center which is still in place. And they also have this Sinberg, as you all might know, that we have this IGM competitions was started around 2003-4 and in US. And now there are a lot of teams from India who also do very well in these competitions where you can engineer organisms for a useful purpose. And this was very important around the same time was to make a registry of standard biological parts. That means I can go and I can order a promoter which I know will work in E. coli. And it has to work on a given amount of output. So there are protocols. The people who were working on synthetic biology or even normal biotechnology, they were sharing different protocols, different genetic parts, and this even till this day, one can order from IGM. So this was a kind of a very nice thing because then the whole community in the world was sharing their own knowledge and exchanging genetic elements. So around 2005, it was something I want to highlight was called something called bacterial photography. So this is a bacterial plate, a Petri plate, where a picture of Einstein can be seen. And how it is done is very simple. That they first engineer bacteria which can sense light. That means there is a sensor which gets active in the dark, and if the sensor is active, the circuit is made in such a way that the bacteria makes a lot of black pigment. However, the moment you shine red light on those areas, the light sensor will become inactive and the black pigment production will go down drastically. So just by controlling the intensity of red light or switching between dark and red, one can have a shade of gray to black to white, right? So that means if we are these places, that means have a saturating amount of red light. However, these places probably have not seen the red light. So they make a lot of dark pigment and you can have a whole shade. So this was called bacterial photography and this was done like 15 years back almost. So this was published. So this also says that that means we can make ingenious cells and control their behavior just by light, right? Then around the same time, first RNA-based regulation of genes were established. So it's very simple, straightforward idea is that if you give an input, then there is a sequence in the RNA which will block its own expression, right? And thus the protein will not be made. However, if you give another input, then a transactivator sequence is made. This sequence will go and bind to this RNA, right? And then it will open up this activation domain. So now the ribosome can bind and it can translate. So the GFP can now be made. So how the cell will behave in absence of this transactivator, there will be less of GFP. However, the moment you add transactivator and its concentration increases, you will have an increase and then it saturates, right? So that means we can control GFP expression by an RNA sequence. And how are these important? So these are called riboregulators, and they are now very widely used in a lot of devices, like biological devices. The idea is simple, that this ribosome binding site will be sequestered by the cis elements, and as I just mentioned, a trans-activating RNA will open it up and the gene is active. And currently in use is something called toehold switches. So there is a small piece of RNA that you can throw into the system, which is complementary to this part, and that will denature this stem loop and thus switch on gene expression. And these have huge therapeutic potential. Currently, there are toehold switch-based synthetic circuits and diagnostic kits. So there is actually a technology now for even for SARS-CoV where you can have this RNA, and they've designed this toehold in such a way that for diagnostic purposes, if the RNA is now, like if the virus is in the blood, you can denature, take the RNA, hybridize with this reporter, and if you see a signal, then you know that you have the virus in your blood. So this has a huge potential in terms of diagnostics as well. Now, as in the 2000s, what also happened, which was the golden age of synthetic biology, that people started to realize that biological systems also work like electrical switches. So they have logic gates, right? So these are some of the simple ones I have put here. AND gate, that means only the output will be there if both A and B, two different inputs, will only lead to output. So these are the truth tables. Only if A and B are there, then you have a yes. So it's a kind of binary switch, right? 0 or 1. NAND is like not AND gate. So that means it's just the opposite of this. If both are there, this output is off. But otherwise, the output is on. So this is the output we are looking at. OR means either of it is there and the gene is on. If both are there, then also it is on. But no input, no output, right? So that is the principle of these switches. So I will just kind of describe what has already been done and in terms of how to design such gates in biology, because these are important to control the cell in a synthetic manner. So let's think of like two inputs. These are again based on all inducible promoters, right? So we have to always keep in mind that synthetic biology uses a lot of inducible promoters. So you need to work with the inducible promoters and the inducers. So arabinose will go and bind to an arabinose response element, which will bind to a repressor. IPTG we know, we inactivates the lac repressor, which goes and bind to the lac operator sequence. Now, in an AND gate, as we just mentioned, both arabinose and IPTG can lead to an output. In this case, a GFP. Now, how to do that? So we can clone a GFP, for example, which is under a promoter, which will only switch on when the transcription factor is bipartite, is in two pieces. They both have to come together to be able to switch on that gene, right? Sorry. So then what people have done, they have cloned one part of this transcription factor under arabinose control, one part of this, the other part of this transcription factor under the lac repressor control. Now, if you have this situation, then there is no output because these two repressors are not being translated. So if they are not translated, then the gene is switched off. However, if you add arabinose, then you are switching off the arabinose repressor. You are switching on the expression of this particular SIC A, but this part of the pathway is still off. So you still don't get an output, right? So if you add arabinose, no output. If you don't add, then both repressors are sitting, no output. However, if you add now on top of that IPTG, then you get an output, because now our IPTG will deactivate the repressor, arabinose will deactivate the arabinose repressor. You will form these two transcription factors independently. They come together and switch on the gene expression. So this is a very simple example of something called AND gate. That means only when input A and input B, only when they are there, the gene will switch on. It's like an electrical circuit. So in principle, these can be integrated to make higher order circuits, and that is a backbone of synthetic biology, because these will depend on, so you can control the expression of a gene just by adding an inducer from outside of the cell. So I'm not going to scare you or something, but I'm just wanted to show that these gates also have been combined to make different gates. So in this case, you have a combination of and NOT gate. Then people can make layered gates. So you can think of all kinds of like a electrical switchboard in a way. So different inducers, if you add, they will switch on different genes. Those gene products will go and interact and finally feed into a reporter. So this reporter you can change into, let's say, a therapeutic molecule. So this therapeutic molecule will only be expressed when it sees all of this as an inducer. So this has a huge implication in terms of biomedicine, these synthetic circuits. So I'll give a very nice example which I found, well, like it was again done in 2010-ish, is that they engineered a E. coli cell which can kill Pseudomonas infection, right? So the Pseudomonas bacteria, it causes infections, and there are some drug-resistant Pseudomonas strains available also. So what this group of people did by using synthetic biology tools, they engineered a bacteria so that the Pseudomonas now has some quorum sensing molecules. It secretes certain chemicals, which is specific to Pseudomonas. Now these chemicals will be picked up by this E. coli, a recombinant E. coli, and that will switch on a gene which will go and activate a gene which will make an antibiotic which kills Pseudomonas, right? But you have to see that once this antibiotic is made, it is within the E. coli. How is going to be delivered to the Pseudomonas? So this antibiotic synthesis is dependent on this chemical secreted by Pseudomonas. But for delivery, this antibiotic, if the concentration of this protein is high, it will lead to lysis of E. coli. So the cells will kill itself, lyse itself, and this antibiotic pyosine is made that will be locally given to the Pseudomonas. So whenever this E. coli sees Pseudomonas, it will make the antibiotic, lyse itself, and kill the Pseudomonas infection then and there itself. So this is a kind of a nice example how these modular synthetic circuits can be used for killing pathogenic bacteria. So yeah, this is just a slide to show that these kind of systems are currently being used and developed. So one can have sensors. We know digital mode is always 0 and 1, right? Either on or off. But biology is not on and off. It is a sort of analog. So there is a whole range of values that we can get. So based on what kind of actuator we have, we can control what kind of output we get. So what chemicals we put in and what kind of therapeutics we add will base on what kind of circuits we design within a cell, let's say bacteria, which will optimize the amount of production of these high value chemicals and pharmaceuticals. Okay, now I will just switch gears for the last 10 minutes or so, is to take you to how synthetic biology approaches has been used for cell-based therapy. Now, in 2016, there was a report of where they engineered receptors on a cell. So we know Notch receptor, which is a classical receptor in biology, which is used for this Notch signaling is used in the developmental processes and many other cell-to-cell interaction. So in a Notch system, there is a sender cell which sends the signal and receiver cells where all these genes expression happen. So the sender cells has a delta, it interacts with this Notch receptor, there is a cleavage, and the intracellular domain of this Notch receptor is a transcription factor. So only upon cleavage, this factor will go in and bind and switch on genes in a Notch dependent manner. what scientists have done in 2016 is that they made a synthetic Notch receptor. That means delete all of this. We can clone a antibody domain which will recognize a ligand, or even a receptor domain which will recognize a ligand, but keep the rest of the notch same. So that means when this ligand and receptor or an antibody antigen interaction happens, there will be a cleavage. Once there is a cleavage, there is a transcription factor which will go and bind to your gene of choice, where you will have cloned the DNA sequence which binds to the specific transcription factor, right? So you can switch on a gene of your choice based on adding a ligand or an antigen. So that means you can design cells which will have a new environmental input, because you can change this, and based on this you have to engineer that, and you will have a novel transcription output. These genes will be switched on based on this transcription factor, which is active only when it sees this interaction. In absence of this interaction, the transcription factor is in the membrane, it is not active. The moment it sees this, this interaction happen, there is a cleavage, and then it can go to the nucleus. So this is very simple, very straightforward, and has been used, now being used for clinical purposes. And how it is done? So in principle, this transcription factor is GAL4, as I was mentioning earlier, and you can have a UAS sequence here. So let's say CD19 is the one which is present in most cancer cells, right? So, so one can design an anti-CD19 Notch receptor. This is a completely synthetic receptor. When it sees a cancer cell, this interaction happens. GAL4 can go into the nucleus, and now instead of a reporter, you can actually clone the cytokine genes, and these cytokines will be secreted by this cell and locally kill this target cell. Right? So there is a beautiful system where it is dependent on the receptor ligand interaction, and people are now working even to make antibodies. For example, you can engineer such a cell with the SynNotch system, where now this gene you can put anything. Instead of a cytokine, you can clone an antibody gene. So upon seeing a signal, this cell will start making their antibody, and one can make anti-PD-1, CTLA4. These are immune checkpoint antibodies. So these are used for many blood cancer therapies and all. So in principle, one can take these cells, add a ligand, and then on a very regulated manner make these antibodies which will go and bind to these target cells, which can be cancer cells, and thus inhibit them from proliferating. So this is all being shown as a proof of principle, and work is ongoing in terms of putting it in the clinic. Now, again, coming back to this golden decade, what another event which happened, another advancement which happens, is metabolic engineering. So metabolic engineering is nothing but we know a metabolic pathway and we change the genes as per what we want the product. So what the first report was there was to make a precursor for artemisinin, right? So this is an anti-malarial compound. Now what people did is that they first did it bacteria, but around 2005 they showed that you can transplant this entire pathway, because yeast has, yeast makes still FDP and squalene. So they modified this pathway by cloning these enzymes, putting in the yeast, and that yeast now makes artemisinic acid. And this artemisinic acid is then purified, and then 1 or 2 steps it makes this antimalarial compound. So that means we don't need this to grow hundreds of acres of plant, and we are saving the biodiversity and the natural resources, because yeast can be fermented in hundreds of liters to make this. And currently, actually, a lot of artemisinin is made in yeast. So how to do that? This is just a picture. You don't have to, see too much into it, but these are how the gene cassettes are arranged. The promoters are cloned, and these are the pathway that one needs to know. And then you engineer in such a way that you finally make an artemisinin, which is a precursor for the antimalarial compound. So now this is the original plant from where the artemisinin was extracted, but now we have the yeast which makes the same things. All these capsules and tablets that is made is actually yeast derived. Not only artemisinin, now yeast has been engineered to make opioids, other kind of like even insulin, because this is the therapeutic protein, and other metabolites like carotene, which is like vitamin A precursor, Resveratrol is good as an antioxidant. So all these high value metabolic products are now being made in yeast, but not from the natural resources. Right? Even the yeast has been used, is a workhorse for synthetic biology purposes to make wine, right? So yeast definitely ferments sugar to alcohol. Can we make a wine which has a flavor of raspberry? So it's a kind of very synthetic biology, very customized application. So all one has to do is to know that yeast has all this pathway of converting phenylalanine into fumaric acid. All you have to do is to clone a couple of enzymes which makes this ketone, and you put this recombinant yeast in the grape juice, and the wine that comes out is now having a flavor or a smell of raspberry. So these are kind of applications of synthetic biology for useful purposes. Now, in terms of looking at gene switches as well, one has, so around 2006, what was also done was to engineer bacteria. So in hypoxic conditions, these bacteria are switched on, otherwise they are just not active at all. And hypoxia comes in tumor environment. So when there are a lot of cell proliferation, the oxygen runs out and hypoxia condition comes. This hypoxia will trigger a gene expression so that these bacteria can now invade these tumor cells and deliver a therapeutic molecule like cytokines or any drug. So this also has been tested in a cell line assay, and now the work is ongoing for use it for medicinal purposes. Okay, now last 5-10 minutes, 5 minutes or so, I'll just focus on what are the other technologies that has been made. So sequencing we saw, which led to the genome read technology, but now manipulation of genomes led to the genome write technology. Now, how to do that? One of the technologies that came is called golden gate assembly. So we can take multiple, almost 24 to 30 DNA inserts, mix them in one tube, and you get this fantastic recombinant plasmid assembled without a problem. And this technology is now widely available. So you can now not only clone 1 or 2 genes, you can do a multi-path gene assembly without worrying about restriction site and other ligase, etc. So you can use this method called golden gate, and it was published in around 2008. In 2009, the other method was published called Gibson assembly, which is again taking different parts of DNA and putting them cloning into a vector in the orientation that we want, right? So this revolutionized cloning business. So artificial DNA now doesn't have to be cloned in a restriction enzyme manner, which is sometimes really painful. But now you can do seamless cloning, scarless cloning by using these two technologies. And using this technology, the first complete synthetic genome of Mycoplasma was made. And this is around 582,000 this base pair genome. It was completely made synthetically with just DNA oligos assembled by using these two technologies. Recently, like in 2014 or so, Professor Chandrasekharan, who is from Pondicherry, actually he is in Johns Hopkins University. His group in collaboration also synthesized the yeast genome, a completely synthetic yeast genome. And the way they did was to just design the whole thing in a computer, synthesize hundreds of DNA fragments, line them up, clone using the Gibson assembly and other sort of things, clone it in a vector, and then replace the yeast DNA, like the chromosome, piece by piece. So now you can have a completely synthetic yeast chromosome, and this work is ongoing, and it's really fascinating work where they want to make a synthetic yeast where you remove all so-called unnecessary parts and make it a biotechnology workhorse. Okay, I'm not going into this, but I will just briefly touch upon. So this is about CRISPR. I think I will just skip this because there may be other speakers who will be speaking about that, and you all know there is a Nobel Prize given this year. So I will just skip all of that for the sake of time. I'm pretty sure you all know what CRISPR can do. Okay, I'll just skip. So these are some of the examples how CRISPR can be used for making genome engineering and modified organisms or curing diseases in a kind of synthetic manner, right? Because these engineering is not natural. We use CRISPR as a tool to engineer organisms. So we can make animals which are immune to HIV. We can in potentially cure human diseases like beta globin thalassemia, and this was quite controversial where they first did it in the human embryos. We can use these tools for diagnostic purposes, where we can distinguish between dengue and Zika, for example, and it is done at room temperature and very fast. So these are all published papers. One can make completely like genome modified pigs. Why? Because pigs can be used in future for organ donation. So we need to have the genome of pigs which does not contain virus. So there have been now virus-free pigs being generated. There is another thing called gene drives, which is used to control mosquito population, and thus all mosquito-borne diseases can be maybe stopped in the near future by using something called gene drive. And this gene drive works in a non-Mendelian fashion to inherit genes. And this has already been published a couple of years back, where they're controlling the mosquito population. They're actually killing of mosquito population just by releasing a recombinant male into the wild. And this has been used to control Zika, and even in some places to control Aedes mosquito-based diseases. Okay, I'm just skipping all of this. The main question is, what's next? What's the future? So the future is engineering cells. So I will really urge all of you to kind of see into it, because the point is that how can we engineer cells based on what we know already? It's a knowledge-driven thing. So how can we put simple stimuli on one end, and we can get the output on the other end, which is for us, like bioplastics, biofluids, chemicals, etc. So for that, what we need, we have to combine different fields of synthetic biology, as I just discussed. We need to have be able to do metabolic engineering, organelle engineering. We need people from synthetic, this systems biology, structural biology, and this all will give, kind of have to come together to engineer a cell reproducibly. And this is a highly data-driven process. So we need computational biologists as such. And just to emphasize, the synthetic biology needs all these people, and that's the intersection of all of this, that this field can go forward in the future. It can solve a lot of problems in this world, and I'm pretty sure that synthetic biology will affect or will, play an important role in all walks of human life and environment, climate change, and even bioeconomy. So I will just show what are the prospects in terms of economy. Nowadays, there is a buzzword where people talk of something called bioeconomy. That means we want to move away from fossil fuels, and we want to use cell as factories to make the compound, the chemical, the protein that we need. So it has already been kind of established that there is a huge market for that. So in 2020, roughly there are around $30 billion. It's a market which is growing exponentially every year. There are a lot of startups now in India and abroad which use synthetic biology-based tools for all these different sectors which are now currently in place. There are a lot of big companies which are involved into this. What has happened from 2011 onwards, when all these tools and etc. were in place, there is an exponential increase in mostly, you can see, bioinformatics based approaches, because it is data-driven science, and also gene measurement and modeling. So these are all in silico. So we need more people to come up with kind of computational or maybe interest in these computers to process this data which is already out there and make hypotheses which the experimentalists can test and engineer cells. So the key technologies in the coming decade or so, or maybe less than that, will be DNA synthesis. So we should be able to synthesize DNA. No more sequencing. I mean, of course, sequencing technologies have reached its peak, but we should have the technology to make DNA in the lab is still quite in its infancy. So the DNA synthesis is going to be a major player. Genome engineering and all these biofuel technologies, pathway engineering, are the future of this field. So one can, for example, and these are all future prospects. A person has a disease because certain metabolite is not present. Can we engineer using a synthetic circuit and then transplant in the tissue of the cell? And only when this person takes a drug, which is an inducer, will switch on this circuit. And thus, this person can become healthy. So these kind of approaches are currently there for different metabolic disorders, monogenic disorders like hemophilia, thalassemia, and sickle cell anemia. So this is currently work in progress. One can also stop infections like cholera by making this gene switches, where a good bacteria, once you take it, like a probiotic, can remove the bad bacteria from the gut and thus cure a disease. Right? Okay, I'm skipping all of that, but there is another thing that is kind of a near future application for personalized medicine is that insulin, right? So we can have a synthetic fat cell where there is a circuit embedded or in the genome where only upon seeing a high glucose, it will secrete insulin and restore the glucose levels. And if there is a less glucose, then this circuit will switch off. So such kind of synthetic adipose tissue can be implanted into an individual which will maintain the glucose and the insulin levels in an auto-regulatory manner. So this idea is also currently there for clinical applications. One can use simple circuits for biosensing. That means we know that there is lack of potable drinking water. So can we have a yeast which we can add into the water, and if it changes color or something, then maybe it has cholera, and maybe if it is not, then it is safe to drink. And we can, since yeast is a safe organism, it will not harm us as well. So there is efforts to make such kind of biosensors for water quality. And work has started, but it is still in the initial stages, is something called biocomputing. So once we understand the different metabolic pathways, which regulates what and all in a cell that happens, and this will come from gene expression data, metabolomics data, proteomics, etc. One can make completely synthetic pathways, something like in a computer motherboard, right? All these genes. So I will end by showing this last slide, which kind of summarizes all the future applications of synthetic biology. And I say it is a fast evolving field, rapidly growing field with a lot of potential in India and all over the world. So it will involve bioengineering, making a proto cell, a completely new cell. And we need some computational biologists in the synthetic biology. Genomics and unnatural molecular biology will also play a big role. And they all have to interact with each other, these different fields, making it really multidisciplinary and really fruitful collaboration between different scientists. So with that, I will acknowledge the group that I have in IVB. So the IVB Synthetic Biology Group is funded by the Department of ITBT, Government of Karnataka, and I have funds from the Ramalingaswami Fellowship for the projects that I run in the lab. And with that, I'll stop, and I thank all of you for your attention, and I'm happy to take any questions that you may have. Thank you.